So in front of you, these are our Baptisia trials, and they just happen to be pretty much in peak today. We do ask that you not take any pictures in these trial beds today. So just recently, you can see all these little black plastic pots. We just recently put out the 2022 batch of Baptisias, this year's class, but you can see ones year by year as they get older. And there's a interesting process that we follow as far as how we keep track of all of them. When we collect the seeds in the summer, we keep track of the pod parents. So that means what plant did the seed come from? We track that. We have some ISO blocks that we track. We hope that we have contained all of the potential pollen parents as well. So we know what are our 3 or 4 options for pollen donors, and then we collect off of a known mom, a known pod parent. Bill, tell them about the planting process, what we do to get them in the ground and get going in a way that we can keep track of them.
Well, as you can see, there are a lot of pipes out here with little black pots, and Jeremy comes out here, brings plants from the greenhouse. These plants are grown from seeds. They're maintained in the greenhouse for a while, and then in the spring we bring those plants out of the greenhouse and we plant them. We have to have pipes that you see here, PVC pipes. Each one has a label, and the label gives information about parent, gives information about when it's planted. As you mentioned, these are all 22. You'll see these are 22 dash with a number. And the 22, guess what year that stands for? 2022. And then there's plant number. And then there's a name, and the name will oftentimes reference the parents, like he was talking. You've got your open pollinated, your pod parents, your ISO. So you will have a number, and then you will have names. For instance, this one is Blue Bunchkin / White Gold X, and it's an ISO number 08. You've got a series of numbers and a series of names that help us trace who the parents or potential parents were to each individual plant. Now, being that many of these are back crossed or crossed, oftentimes the siblings are going to have a lot of differences. So one of the reasons we put out a lot of plants, and if you look, some of these are grouped together, you'll see that the numbers and the names are oftentimes very similar because they share the same pod parent. And one of the things that we're doing is looking for them over time to look for characteristics or traits that may be accentuations of the parent, may be better. So it will eventually be a culling process. As far as installation, we dig our hole, we shake out the soil, we mix a little bit of the potted soil with the native soil. We make sure that the plant is planted at the same depth that it was grown. We don't want to plant it too deep, we don't want to plant it too shallow. And then we water it in. And of course, depending on sunshine and temperature, that also may depend on how often you have to water it. We want to keep them well watered. We want to keep them protected. We don't want to pull the mulch up over them. We want to keep the mulch over the soil. And we want to watch them every day. We look for wilting, or we look for any potential problems that may be associated with planting issues.
Yeah, that is right. So there's a lot of the back end, a lot of the maintenance of these plants. However, in the garden, they're one of the most maintenance-free, one of the easiest plants that you can grow in really dry soil, really wet soil, sandy soil, clay soil, cold places, hot places. They pretty much grow everywhere. And all of these genetics are originally derived from original wild collections made mainly in the 90s. So you're looking at over 20 years of generally open pollinated selections. So this field, its purpose is for breeding and the evaluation of breeding. And there are different reasons for having certain fields, which I'll get into. We'll get into that a little bit later. So the reason for having an ISO block, and I probably can't say it as well as Tony can, but I can say that seeing that this is a seed from white gold and it is teamed up, it is isolated with blue bunchkin, he's probably looking for yellow foliage with a purple or blue flower. That would be my guess, and probably something a little smaller than others would be my guess as to where he's going. So there are certain phenotypic, aesthetic, whatever you want to call it, umbrellas. And you want, as you pursue a breeding program, you want to try to have real goals in mind for what you want to produce. And an ISO block is still an easy, low-cost way to do that, but it is a very focused way to breed plants. And so we're getting there. We mix the genes up really good for the first decade or so, and then we settle them out. We decide where we want to go with it, and then you get this Baptisia trial. So I would say a little over half of the Baptisias that are in House 4 for sale today came from this field. Blue towers, ivory towers, yellow towers, blonde bombshell, and there are several others. 3 of them are patented now. We dig up several plants every year out of this field. Over 200 Baptisias we will dig up and destroy because they didn't make the cut. And that's the other side of this. So there's the planting side, and then there's the brutal selection side. And I don't see any new red flags today, but last week we dug up about 75 Baptisias and destroyed them. There's their carcasses over there, actually.
So in this whole field, you're just making new ones?
We are correct. Yes. So we're not, this isn't, you have a Baptisia breeding program. We're not taking your Baptisias and trialing them out here. We're taking Baptisias that we collected and grew from seed out here. And so yes, we're making new ones. So that's what we're doing. This is purely production. Yes, it's a native plant, but we're not doing it because it's a native plant. We're doing it because they're very beautiful. There's so much beautiful aesthetic garden potential here. Yes?
Jeremy, where are you doing the isoblock though? The isoblock's not here, is it?
They're not, no. We have a couple acres up 500 feet that way. We try to keep the isolation blocks at least 40 feet, 50 feet apart. And it's not a perfect system. But at that stage, they're in a 1-quart pot, they're sitting in greenhouse 16, and they're in the 1-quart flats. Tony will go in there and he'll look through them, and he sees these are seedlings of an isoblock. And he can see, especially if it's something easy like we want yellow leaves. Okay, does it have yellow leaves? And I mean, yeah, that's pretty easy. Occasionally they'll throw a flower, but it's more, is it vigorous at that stage? Because we want vigorous plants. People don't want to have to wait 10 years for 20 stems. They want 20 stems in their first year. And some of these Baptisias can actually come through with that. So he's looking for things like that to make sure that yes, it's following the direction we want to go. Even before it flowers, he can tell. Or it's just going in a completely crazy direction. Hey, you gotta check that out and see if it actually, what's going to happen. And then he also comes out here now that they're blooming and he says, I like that. I'm not going to put any flag on it at all. I don't like that flag. And red flag, it gets dug up immediately. So some Baptisias will stay out here without ever any hope of being released, but they won't ever get a red flag because they have a desirable trait, something that maybe will come through their pollen, or maybe we can continue collecting seed from them. And we'll get something where we want to be. And when we do, we'll probably then red flag that one. So it's brutal, but that's the only way really to have a truly groundbreaking, no pun intended, breeding program like this one. So research, trials, outdoor production, Bill and I do all of that. Back here, we'll switch gears a little bit. These are one of our 2 Crinum fields. So we've got a bunch of Crinums here, a bunch more Crinums over there. What you're looking at, there's like 3, 4, 5 different things going on. This is a breeding program. We collect our own seed. We make some intentional crosses. But we also, like we were saying, here there aren't other people's genetics so much, but here there are. So we have about 200, 250 of other people's clones out here. And what we're able to do is, especially in something like Crinum, a true pass-along plant. It never dies. And so there's a lot of sorting out to do in the genus. A lot of this clone and that clone are named something different, but they're actually the same thing. That happens a lot. And so we have this in part as a reference collection. We've got all the players in the room and we can just look at them and say, you're different. You're the same as that one. I didn't know that, but actually you're also the same as that one. This one was thought to be that one, but it's definitely not. It's amazing. It doesn't take necessarily a PhD or even a master's. the way that you can set up a trial can make the results manifest themselves as some very low-hanging fruit, and it works out great. It's not difficult or high cost to maintain, but the results are there. Seedling evaluations, again, we collect Crinum seed, we grow it. We have a trial where we row them out until they flower the first time. Then we red flag some, we blue flag others. The blue flag ones come out here. We give them their own social security number. They've achieved ascension. Doesn't mean they're saved from red flags forever, but that's the next step. Mahonias, again, this is open pollination. It's just like the Baptisia, except they're Mahonias. You can see a bunch over there. There's a bunch over there, a couple 100 feet. So we've got about 600, 700 Mahonias out there right now. In that super secret white studio over there are our Epimediums. We trial other people's Epimediums. We come up with our own seedlings, open pollinated again. So there's a lot of cool stuff that comes out of there. Not enough room to take the tour in there, but you can see a lot of the results of that breeding in House 6 today. Huh? You can peek in there, yeah. Again, no pictures, but you can peek. There's no way everybody's going to fit in that house, but yeah, if we would like to walk there, we can, certainly. Let's walk that way.
So once you have found a new variety, I don't know the technical terms, but that you like and would like to carry forward to give it the social security number and name it and perhaps produce it, how do you keep it pure so that it doesn't get...
Okay, well,
does that make sense?
it does in the sense that right now I'm standing amongst other people, yet my DNA remains the same. So the same goes there. Once they're a plant, they're a standalone plant. We're not looking to, we will then propagate from asexually, probably from divisions, things like that. Occasionally you have seed cultivars. Yeah, we'll take a right here.
How the cross-pollination and all of that doesn't come into play anymore?
It doesn't affect the plant as it is. Again, I'm standing amongst other humans.
How do you propagate it?
Divisions in most cases. The Baptisias we take from cuttings. So we take a piece off of it with the pruners, and in the prop shed they work their magic and they make more of them. Take a left.
Yeah, that's how. I was
Yep.
wondering whether, because how can you control which a bee? You can't.
Bill, you want to tell them more about these? And I don't know if you've collected seedlings
I
yet.
have not collected seedlings on Epimediums.
I'm happy to discuss that if you want to.
I think I'll let Jeremy talk about collecting seedlings, collecting seeds with Epimediums. But Epimediums they're really, really cool plants, Asiatic. They're related to Mahonia, related to Nandina. Much lower growing form, and the flowers are much more intricate. And we're going to let you kind of sneak some peeks in here. The flowers are, I'd say for most of them, kind of coming to an end. A few weeks ago, they had climaxed, really impressive.
Yeah.
We cover it with the frost cloth a little bit earlier than we do with a lot of the other plants, because what Tony wants to do is try to get a pretty close to synchronous blooming with many of these, because he likes to photograph them. So since Epimediums oftentimes are a really early plant, they start emerging early. We want to protect them from freeze because we want to make sure that the flowers develop. And at the same time, like I said, we kind of want to accelerate the blooming so that Tony has his more what would you call it, condensed window for being able to
fewer trips.
photograph.
Yeah,
fewer trips.
Yeah.
And the shade cloth, like we mentioned, perfect for diffuse light. Epimediums are an understory plant typically, which means they don't like bright sunlight. They do tend to burn. They do tend to get damaged. So here we have the shade frost cloth house for Epimediums, and we have had to do supplementary heat occasionally on really cold nighs. So sometimes we have to bring in bullet heaters and monitor the temperature. So a couple times this spring we already did that. So I'm going to step aside and just let folks peek through these doors here. We do ask, please do not take pictures of plants. And we can pretty much, if you guys want to, just kind of different people spread out a little bit, peek through these doors, but you'll see that there's, I'm going to let there you go. So lots of epimediums, and many of them are white, some are pink, coral colored, yellows, epimediums. They come in a lot of different colors. Some of my favorite ones are I like some of the kind of creamy pink. There's one that's like flamingo or something.
Yeah, there's pink flamingos.
Pink flamingos.
I think, yeah.
I'm from Florida, so hey, I like pink flamingos. But no, they're actually quite incredible, and they're really pretty plants. The leaf structure. These are some epimediums behind you here, so you can see just the foliage. They're also a nice understory foliage plant. Not quite a ground cover, but kind of close.
These are. I mean, these run.
These do run,
They're
yeah.
up there because they're going to paint that hillside.
Can't survive a male dog. A big one.
You know, some of these runners, if the dog stands still long enough.
Hosed it down.
Oh yeah, that
After
does.
5 years.
Yeah.
Get a female dog. It's less of an issue.
Yeah. God bless him. He's fine now.
But
I'm sorry.
No, we love him.
Do you cut these back in the winter?
we do. We like to. There are deciduous epimediums, and then there are evergreen epimediums. We like to try to hold on as long as possible to our evergreens up until they begin to put their new growth up, for a couple reasons. One, we get to monitor just how evergreen they are. You know, if we have one of those single-digit years and those leaves are still green or they're purple or whatever color they're going to be, but they still look good, then we can put that on our metal signs when we're selling them. But also, by leaving the foliage up, that's going to shade its own crown. And by shading its own crown, it's going to wait a little longer. Think something like Epimedium whooshinense. That thing wants to come up in February every year, and so you can bet it will get frozen. And so if we can let it hold on 1 or 2 more weeks before it really pops, then by then we'll cover it with the photo studio. Because it's behind me, the weakness of this structure, outside of the fact it's old 20-year-old 4x4s, is that when we have the photo studio up, any snow at all, we've got to take it down. We've got to drop the ceiling. We've got to do that so it doesn't crush itself. And so we need to wait as long as we can to put the photo studio up. So we want to delay the plants coming up, but once it's up, we like to have it warm. Plants come up quickly, and we get a beautiful flowering. Not just the light, but it also kills the wind. And epimediums are one of the hardest plants to photograph. That's what Tony always tells me. And then I tried taking a picture of them at my house, and it's really difficult. So, yeah.
But in nature, they're hardy.
Very much so. Yeah, very much so. The common name is horny goatweed. I can't imagine why it's called that.
Chinese medicine.
Yeah, Chinese medicine. That's
That's the translation.
Yeah, that sums it up. But so we've got several different trials, and some trials are for breeding, others are for truly just evaluation. Some are a reference collection so that we can put everything side by side and get to know it better. We believe at our very core in conservation through propagation. And so, you know, when we're evaluating something, we want to, if it's a very rare plant, let's see what it can do, and let's put ourselves in a position to make more of that plant so that it's not as rare, it's not as threatened. And so one way that we can do that is through our outdoor production program. Beautiful garden plants that, very rare plants in some cases, that can be divided or that we can take cuttings from. We can make more of those. We can grow them in a trial situation so that the maintenance is limited, but they present themselves for efficient propagation. And actually that's something I think most gardeners can do at their own garden. If you have one little bed that you more or less grid out, or you put certain kinds of plants in areas where you know you can get at them with a shovel to share with your friends, it's not too difficult. But you'll see a lot of our stuff in research and outdoor production is gridded out, which helps with the maintenance cost and that sort of thing. So we've got a lot of other research and outdoor production areas. I'd say come to future open houses. If one of us in the staff shirt can give you a tour, we certainly will. But that said, do you have any questions for Bill or I? Yes?
Here, do you do more open pollination or manual pollination?
Mostly open pollination. And that's just because it's difficult necessarily to have someone there all the time ready to make hand crosses. We've got a lot of cross-pollination controlled going on in the podophyllum right now. Our garden curator, in fact, is doing a lot of hand crossing. Tony Avent, myself, and a few other people have helped out with the Erycema, Jack in the Pulpit. We've been doing a lot of hand crosses, coming up with some incredible interspecific hybrids there. Oh, what's in our pipeline is really exciting for Jack in the Pulpits. Other hand crosses, hostas. We've been hand crossing hostas since I think 80. Well, our breeding program's been going on since 1984. Yeah, that's true. Agave.
A lot of agave.
Yep.
A lot of agave, hand pollinated.
Yep, hand pollinating agave, hand pollinating true cactus, echinocactus, trichocereus, all these different things, and they seem to cross outside of their own genus rather freely. So yes, some hand crossing, mostly open pollination. Even though we rattled off a bunch of things that we actually do hand cross, there's just that much more breeding going on here. There's a lot. Does that answer your question? Okay, cool. Any other questions?
Here's your question.
Yeah.
How vigorous do they expand?
One of the Baptisias that we patented, called Ivory Towers, it is so vigorous that the mass producer, and we patented it, so the mass producer that we hope to make a connection with and really get this thing into the mainstream, their field knives or whatever their more or less automated harvest system, it couldn't handle it. And it was built for Baptisia. And so, Ivory Towers, a lot of our stuff, it's bred to be extremely vigorous. You buy that 1-quart pot with 1 or 2 stems, and in good conditions, good garden conditions, in 2 or 3 years you could have 40 or more stems.
Oh, Lord.
It's incredible what the potential in Baptisia is, and it only gets better. There's a couple Baptisia out there that in 2 years have 30 to 40 stems, and that's from a 1-quart pot like you saw.
You do have types that aren't quite so, like
We do.
I don't want to fight to control.
Well, and they're not runners, and that's the other thing. Well, there are some that do kind of run, and they get a red flag pretty quick.
Okay.
We want tight clumping plants that are dense. So 30 or 40 stems, but those 30 or 40 stems are actually in this much space. They are coming out of,
It's not going to, because it comes up on a stalk that looks like
Yeah.
Solomon seal. My Solomon seal has been very clumpy.
some are clumpers, some are more runners.
This year,
Yeah, it depends on if the crown was messed with in some way. Sometimes that can trigger it. Yeah, they say it's impossible for 2 objects to share the same space. I'm not sure. Some of these clumping Baptisia might break that rule or bend that rule of physics in some way. But we want tight clumpers, and we want a lot of stems. We want vigor. We want it to look good for you as soon as possible. Yes?
My question follows up from part of that, which is you were talking about how, I think you said 3 of your Baptisia are patented.
I think currently we have 3, yes.
So you create lots and lots of different varieties. What goes into deciding what gets patented, or what's the... Talk a little more about that.
Recently, I've got a little experience personally now with patenting a plant. But what I understand is there's about a $3,000 to $4,000 cost per plant to patent. So you're going to get a royalty on it for 20 years. So you want to make sure that you can make money on that, and quick too. You don't want to blow through 3 or 4 grand and sit on that deficit for 10 years. So part of that too is then there are certain companies who will help market and help mass produce. They basically facilitate somebody making money, and themselves included in this case.
So the patented ones are the ones that are the best candidates for some other place to mass produce and
Yes.
sell at a
So
grand scale.
sometimes, the fact that we've got 3 patented Baptisias, that's not to say that those are absolutely the 3 best. They're simply 3 Baptisias that other mass producers seem to think that they can really take off with, and that it warrants protecting so no one else can patent it without paying them money. And in doing so, you have to get permission and get a license from them, or get permission from them, from the patent holder, to even propagate it, royalty or not. So that's really what goes into a lot of it, whether or not it's a beautiful plant. If we've got this gorgeous Baptisia, but it's probably not going to hit the big time, we're happy to not patent it, not spend anything except to propagate it in-house. Other people can buy it. They're paying, $18, $25, $30 a pot. So we're making our money back for us being out there, for Bill and I being out there weeding, mulching, digging up red flags, planting new pots. There's a lot of money that goes into this, and that's why we try to keep the cost as low as possible, even including on our 1 quart pots being sold. It's simply, there's a lot of money that goes into it. The overhead is large. So yeah, we'd love to patent more, and that would just be a good indication that they're just selling that well. So, any other questions? No? Well, thanks for coming out. I tell you what, we can help walk you back to the area